Hi, our team is confirmed by Alejandra Ode Valdez, Ana Lucía Garza González, Alicia Cristina Sánchez Flores, Alejandra Guadalupe Rodríguez Ham, Nancy Fernandez Covardueñez, and Cecia Nabil Montoya González. In this video, we are going to talk about the DNA as this model that I have here, their structure and some discoveries as the inheritance molecule and more. But first of all, what is the DNA? The DNA, as you may know, is the hereditary material in humans and almost all their organisms. Nearly every cell in a person's body has the same DNA. Most DNA is located in the cell nucleus. The foremost job of DNA as a molecule of heredity is to store information. Genes control patterns of development, which means that the instructions that cause a single cell to develop into an oak tree, a sea urchin, or a duck must be somehow be written into the DNA of each of these organisms.
Well, now nucleic acids. A nucleic acid is an important class of macromolecule which it is found in all cells and viruses. And the functions of the nucleic acids, well, they have to do with the storage and expression of the genetic information. And the DNA encodes the information that the cells it needs to make proteins. And a related type of nucleic acid that is called ribonucleic acid, which is the RNA that comes in molecular forms, really different molecular forms that participate in protein synthesis. And the nucleotides, it is the basic building block of nucleic acids. And the RNA and the DNA are polymers made along with change of nucleotides. A nucleotide consists of a sugar molecule, either can be ribosome, RNA, or deoxyribosome in DNA. It is attached to a phosphate group and a nitrogen-containing base.
Now we're going to talk about the nitrogenous bases and the covalent bonds in DNA. Cytosine and Y9 bond-based pair, they have 3 hydrogen bonds between them. Adenine and thymine have only 2. This extra hydrogen bond helps make the cytosine Y9 pair favorable because it increases the stability and reduces bond energy. Ionic and covalent bonds do not occur in nitrogenous bases in DNA. Covalent bonds are found in the DNA backbone, known as the phosphodiester bonds.
The chemical nature and structure of DNA were not elucidated until the middle of the 20th century. Prior to that point, scientists had spent years speculating about which of the many types of molecules within cells contained the hereditary information. Eventually, researchers zeroed in on DNA as the substance responsible for the transfer of traits from one generation to the next. From there, the race was on to learn more about this remarkable molecule. Although James Watson and Francis Crick determined the double helical structure of DNA itself was identified nearly 90 years later by Swiss chemist Friedrich Miescher. While studying blood cells, Miescher isolated a previously unknown type of molecule that was slightly acidic and contained a high percentage of phosphorus. Miescher named this molecule nuclein, which was later changed to nucleic acid and eventually to desoxyribonucleic acid, or DNA. Miescher did not believe that nuclei was the carrier of hereditary information because he thought it lacked the variability necessary to account for the incredible diversity among organisms. Rather, like most scientists of his time, Miescher believed that the proteins were responsible for heredity because they existed in such a wide variety of forms. For multiple decades following Miescher's discovery, most scientists continued to believe that the protein, not DNA, was the carrier of the hereditary information. This changed in 1944 when biologist Oswald Avery performed a series of groundbreaking experiments with the bacteria that caused pneumonia. At the time, scientists knew that some types of these bacteria, called S-type, had an outer layer called a capsule, but the other types, called R-type, did not. Through a series of the experiments, Avery and his colleagues found that only DNA could change R-type bacteria into Si. This meant that something about DNA allowed it to carry instructions from one cell to another. This was not true in any other substance within the bacteria, including the protein. This resulting highlighted the DNA as transforming factor, thereby making it the best candidate for hereditary material.
Watson and Crick attempted to build a DNA model without the help of photograph 51. They organized the molecule in ascending order with strands like railings of a staircase. This is the double helix model. These two strands were formed by covalent bonds between hydroxyl groups of the deoxyribose sugar and phosphate rings. This link is called phosphodiester bond. This model, the double helix model, looks like a twisted ladder. It counts for Franklin's X-ray pattern and explains Chargaff's rule of base pairing and how the two strands of DNA are held together.
The nucleotides are identical except for the base, which can be adenine, thymine, guanine, or cytosine. These are chemical cross-links between the two strands of DNA forming bases held together by hydrogen bonds. They always pair up in a particular way, called complementary base pairing. There are thymine pairs with adenine and guanine pairs with cytosine. These basic units are linked together to form strands by strong bonds between the deoxyribose sugar of one nucleotide and the phosphate of the next nucleotide. These strong bonds form sugar-phosphate backbone. The ends of DNA strands are called the 5' end and the phosphate end, and the 3' end the oxyribose end. And the two strands in DNA are antiparallel, which means that one strand runs 5' to 3' direction, and the other runs in 3' to 5' direction. This creates the twisting double helix structure of DNA.